Welcome to Histology Week 2, weekly review video on nerve tissue and muscle tissue. So I found this picture from your lab manual. It just shows different structures that you'll need to know, terms you'll need to memorize. I talked about in my last video, I went a little ahead of myself, and we already talked about skeletal smooth and cardiac muscle longitudinal and cross sections. So if you want to review that, go ahead and go back to that video from last week. It's in the discussion board. Talked about striations. So basically what we're going to go over in this video concerning muscles is the sarcomere and the structures within those and the layers of connective tissue surrounding the muscles. So this here is the sarcomere. We have the myosin and the actin, and if you're in the lecture, you'll learn how those work together to make your muscles move. But for now, we're just looking at the structures. This here is the Z disc. A lot of people will sometimes spell it with K. Just note that it's spelled with the C. Not a huge difference, but be careful of that. The I band, which goes from the end of one myosin filament to the start of another. So there's no myosin in an I band. And then in the A band, it's from the start of a myosin, or it's the length of one myosin filament. And then there's this little spot right in here where there's only myosin, there's no actin, which is called the H zone. Then let's look at just kind of a picture of what this looks like. So something to help you remember these, the I band is the light band, I band. So that's from here to here. That's an I band. And then the A band is the dark band because it's a thick filament. So it's from here to there. And then this arrow is pointing to the Z disc all the way up. And then this marks the H zone right here. So this is the layers of connective tissue surrounding each layer of muscle. So we start at the top, from largest to smallest, epimysium, paramysium, and endomysium. You can look up the root of each of these words. That'll probably help you remember those. Epi meaning above, I think so, yeah. Peri meaning around, and endo meaning within. So this is a really helpful diagram. Around each whole muscle is the epimysium. Around the individual fascicle is the paramysium. And then each muscle fiber has its own layer right here called the endomysium. So epimysium, paramysium, endomysium. Moving on to nervous tissue, we will go over pretty much all of this in detail since I didn't do that in the last video. You'll want to know, be able to identify a slide of spinal cord smear, which takes basically what it sounds like is a sample of nervous tissue from the gray matter of the spinal cord, smears it onto a slide, and you look at it under a microscope, and that'll show neurons and dendrites and axons, all of that. And then we'll go over a cross-section of the spinal cord. And again, the layers of connective tissue. So this here is a spinal cord smear. You can see that the structure's been disrupted. They're not like connected really. So this is, each of these are neurons. And then this part right here is the soma, or the cell body. And then within the cell body is the nucleus. Sometimes people will get confused when we're asking for the soma, they'll call it the nucleus, or when we're asking for the nucleus, they'll call it the soma. Just be aware of that. In the Atlas review video, it talks about how unless it's really specific and asks you, okay, this structure delivers neurological signals from the neuron to the rest of the body, then you can call it an axon. Or if it says, okay, so this structure delivers neurological signals from the body to the neuron, then okay, that's a dendrite. But unless they give you that information, it's not, you can't really tell which one's an axon and which one's a dendrite. You'd be kind of guessing on that. And then the other thing in this slide that you'll need to be able to identify are all these little dots right here are neuroglia, or neuroglial cells. And then this is a cross-section of the spinal cord. We'll start here with, this was helpful, it talks about this in the video in the Atlas, is that gray matter has neuron cell bodies and dendrites. So it'll look like this. But then white matter has myelinated axons, which accounts for the difference in color here when you stain it. So let's just start naming structures here. This is the anterior median fissure and the posterior median sulcus. So median meaning it's toward the center of the body, and then sulcus and fissure meaning there's like a space, and then anterior and posterior. You'll notice that the anterior is usually more distinctive. It's usually a little bit bigger, so that helps you determine that these are the ventral horns at the front of the body, and then these being the dorsal horns on the back side. And then on the side here are lateral horns. They're not usually very prominent. And then this section connecting the two sides is the gray commissure or gray commissures. I don't know. People say it different all the time, but I call it the gray commissure or whatever. And then this is the central canal. These right here of white matter are the lateral columns. This is the the ventral column and the dorsal column. So sometimes it'll ask you to get specific in quizzes where it says dorsal column of white matter and ventral column of white matter. And then this will be like ventral horn of gray matter and dorsal horn of gray matter. So you can get specific like that if you want to. Let's see. I think that's it for this slide. So this one shows better the dorsal root and the ventral root and the dorsal root ganglion. Also in your atlas video, I would really recommend watching that because it goes into detail or more detail than I will about these structures on the outside here and how to better identify those. So I think that's everything we have here. So this is just a slide, a longitudinal section of nervous tissue. Most everything that you need to know is already labeled on here. But so this right here is the myelin sheath. All these right here, myelin sheath. And then this line going through is the axon. Just stretching all the way through each of these. And then this right here, it comments on the endoneurium, which we'll talk about more in a second, around each individual axon. But then right here, where two Schwann cells meet, is the node of Ranvier. I think that's everything on this slide. So again, just like muscle, there's the epineurium, perineurium, and the endoneurium from largest to smallest. And another picture, just like in the muscle, super helpful. Around every spinal nerve, there's going to be the epineurium. So remember that it's around the entire nerve. And then for each fascicle, there's the perineurium. And then each individual axon is going to have the layer of endoneurium. This is just a cross section of white matter here. You can see all the myelinated axons. Every single one of these little circles is an axon. So like all these, this dark dot right there is the axon, and then the white space around it is the myelin sheath. And then the little outline around each of those is the endoneurium. And this here is the perineurium, but you can't see in this slide the epineurium. So you can see the perineurium, the endoneurium, the myelin sheath, and the axon. This is just a close-up look of that last slide where it has the axon, the myelinated sheath, and the endoneurium. All right, I think that's all I have for you guys. If you have any questions, put them in the discussion with this video, and your classmates can help you answer those. I can help you answer any questions. If you have any other helpful tips you'd like to add to this, go ahead and put those there as well. I think that's it. Good luck, you guys.